That was great. I'm going to pick on Sam just because there's this big thing about fibrosis in muscle, and you're measuring area there. If you measure total collagen content to the muscle, diseased muscle, is that fibrotic muscle actually got more collagen in it, or does the collagen just take up more space?
Well, there's a couple of ways to answer that. So I would say yes, there's more collagen. We've measured it two ways. I mean, trichrome is the easy way. I don't think that's a good way to do it. Hydroxyproline is more quantitative, but also noisy. But those two things line up with each other roughly. And I would say the answer is yes, because we never just end at the quantification of protein. We test it to function, and the muscles are all stiffer.
Yeah, okay. But if you look at total collagen content, even in a diseased muscle, you'll never really get over 3%, right? So
I don't think that's true.
Is it angle or other things? Well,
I mean, it might be true in a small animal, but in a human, for example, like this is an issue about models of disease.
Yeah, exactly.
So mouse and rat, for example, and cuff. I mean, I've done hundreds of experiments in all those model systems, and I can't make a mouse or a rat look anything like the phenotype of a human. Other people can somehow.
okay.
Those are all changes in the single-digit percentages, but if you go in and start biopsying humans, you wind up with sampling that's over 50% connective tissue and fat. Now, even more disturbing is if you stereotactically biopsy a human based on the MR data, and you look for pink, striated, bleeding tissue on biopsy, more than half of those samples don't even have myosin in them.
Right.
That's terrifying.
That component, completely agree.
Yeah. Okay.
No, you must be treating your mice too well.
Yeah, I know. Too nice.
They're luxuriating in their
right.
But even
beds. Yeah.
but even Luke, who's done a lot of the looking at it throughout the age, he can never really find more than about 3% collagen within the matrix, even in old diseased human. But
Okay, maybe we should talk about it and look at quantification schemes.
Sounds good. I'll join that conversation. Next question, please.
Hi, Andy Fry, Kansas. This is for, I think, Annabelle. And this may not be fair because we've had conversations with your group over this past year, but in your model, you're separating out exercise resistance as the stimulus or endurance. How difficult would it be, or what can you tell me about looking deeper into the resistance side? Because not all resistance exercise is the same, different loads, different rates, different volumes, et cetera. Because that's kind of where we get really excited, because there's a lot of potential with that model.
So for the model, the two parameters, the input that can be changed right now is the weight of the exercise, which doesn't really have a specific meaning, but that could be like interpreted as the intensity. And it's like a percentage from 0 to 1. And then the other thing is the timing. So right now, those are sort of the only two variables that can be changed. But I think that eventually there could be a potential to sort of make it more specific, so it could like incorporate sort of more directly like things like the actual weight that's being used, or like the intensity or the type of exercise.
Great, thanks. Next question.
Kang Shan from Stanford. This is a question for Milena. So you showed that you went through this fairly quickly. There seems to be discrepancy between the transcriptomic changes and then the protein levels in mitochondria post-exercise.
Yeah.
Question is, so that suggests that it is post-transcriptional or translational change or protein degradation?
I think it's important. I didn't have time to go through all the details with the short time, but it's important to say that these samples that were from 1 to 4 and 8 weeks or 4 and 8 weeks after training are taken 48 hours after the last exercise bout. So for some tissues where there was a large discrepancy, that could also be due to the fact that transcription usually is more transient. So like in humans, for example, we see that transcriptional changes usually peak around 3-4 hours after exercise. And protein abundance is, of course, something that happens a lot later, usually more 24-48 hours. So that could be one explanation why there are discrepancies. But yeah, there were also a lot of differences between tissues, as we saw. And we also saw a lot of sex differences. So yeah, we can't say everything because we only have the snapshot from this 48 hours after.
Great.
Hi,
Next question.
Yeah, Rocky Twan. Follow-up question for mitochondria. Eons ago, when we first studied cell biology, a mitochondrion in a very active cell on a electron micrograph doesn't look the same as a quiescent cell. It's more puffy. It's the cristae structure is more expanded, and so on and so forth. All kinds of theories came from all those observations. I have a question about exercise and mitochondrial activity. Now, you focus on transcriptomics and whatnot. Definitely that has a reason. But is it possible that it's a structural aspect? Because mitochondria is made up of thousands of little batteries, each of which is the electron transport chain driven by a proton gradient. And all of that is dependent on the nano, sub-nano distances between molecules. And that drives the efficiency of your ATP generating system. Is it possible, because when you exercise, you need to react pretty fast. Now you have the creatine kinase and all this stuff. But the point is it possible there are structural changes within these electron transport units, the 3 ATPs that's generated from the cascade? I mean, is it possible that there's a little bit of separation, or is it more clustering, packaging? Has anybody looked at these things? I haven't thought about this for a long, long time until you started talking, and I started thinking about this.
Yeah, that's
Thanks.
a great question. We do see, obviously there are, latest research shows that they are way more dynamic than we thought before, and there is constant fission and fusion, especially in skeletal muscle and the heart. And we also see differences in, for example, cristae signaling pathways with what we have. I can't speak directly to the structural changes since we haven't done any like electron microscopy, for example, in these, but we're actually discussing looking at that as one of the sort of follow-up things related to the signaling pathways for cristae formation, and look both in our human athlete muscle as well as in endurance trained rats to see if we can dig deeper into how that happens. And then, of course, going down into the electron transport chain level is the next step as well.
And inside the inner membrane,
there's
got to be a lot of things happening. Thank you.
I think so, yeah. So it's
All
very
right.
exciting.
Let's see if we can get 2 more questions in. Go for it.
Hi, Chris Minson, Oregon. So I'm curious on the time-restricted feeding aspect. I know that became really a popular thing to be talking about, and then some of the research more recently doesn't seem to be backing up the efficacy of that for many things like weight loss or other things. But when it comes to an elite athlete, I get really a little bit twitchy about time-restricted feeding, because most elite athletes, they're balancing a lot of things. It could be a lot of them have to get up early in the morning to do their workouts. They need to fuel those workouts. They got to be eating their first meal at 5 or 6 in the morning to make sure they can get their workout in and have a fueled workout session. They're also trying to deal with all kinds of things during the day. They may not be able to choose when they work out or when they train if they're on a team or sports thing. Oftentimes you want to try and fuel their sleep as well, make sure they have some of the right amino acids before they go to sleep for protein regenerating. So how do you really see reconciling time-restricted feeding with the realities of the idealized feeding and fueling and training for an elite athlete?
Yeah, first of all the weight loss, you said that it does not hold up. We can talk about it. I have written quite a few reviews what is wrong in those highly publicized studies. Second, when it comes to athletes, yes, I understand that the goal is how to perform tomorrow. The goal is actually not, in many cases, how to keep the athlete healthy until this person is 50 years old. So when people say, what can I eat to sleep better? And there is a lot of popular media about amino acid supplementation to sleep better. I'm sorry. Those things actually, biologically, they do more damage than keeping the athletes. So we can talk more about the mechanism and how it applies. But what we see is almost everybody who does time-restricted eating, at least stopping eating 3 hours before bedtime, their sleep quality improves. And the acid reflux and gut health actually improves. There is much better rewiring of the gut microbiome. So that may be one indirect effect. And in the morning, I still have difficulty to figure out whether morning intensive exercise is actually good in long run, because you're losing sleep and your motor coordination is really bad in the morning. So it'll be interesting to see whether there are more injuries in the morning than in the afternoon.
Thank you.
All right, we do have special dispensation for one last question. Please, come ahead.
What happens in women who recently gave birth? So do you get a break from the circadian rhythm?
No, actually, so one big thing about women who had given birth is there is postpartum depression. And we always think that is linked to, of course, some of it is linked to the change in life. But many women, they are staying awake all night because they're waking up many times. And then throughout the day, many of them are stuck inside the home. They don't get enough light to reset the brain clock. That's one. And second is, light is the best antidepressant, because those who live in the Nordic countries, they know it pretty well. In wintertime, just getting 10,000 lux of light for an hour is a big antidepressant. Many women don't get that. So it will be interesting to do a study, particularly for postpartum depression, in women athletes, to see whether they can get over with better light.
Thank you.
Great. I'm going to use the chair's prerogative to ask one last question to John, because this morning when I was doing my HIIT exercise, I did not feel hungry afterwards, since I felt almost nauseated by the time I finished. But I think that most of us who would do more moderate intensity aerobic exercise would realize that we have a tendency to want to eat more after, and that can be pretty hard for those trying to lose weight. Can you speak to that a little bit in terms of your feed work?
Sure. So there's appetite is complicated, and there's a lot of different appetite hormones that work at different time scales. Some work very quickly, some work over a long time. What we found with lac-phe is that it's acute, and it's most strongly stimulated by vigorous exercise, like the HIIT that you're talking about. There's going to be other appetite regulating molecules that are stimulated by endurance work. That's a very different type of work than high intensity work. And there are other appetite regulating molecules that are going to kick in over a longer time scale, maybe several hours after you work out, compared to sort of the acute time frame. And so there's a lot of molecules left to discover.
Yeah, but we're glad we have you to measure them. All right, please join me in thanking the panel.